Good morning, guys. Welcome to another instalment of Biology from Home with Miss Cherry. Yay! Today we're going to carry on and we're going to keep going through this genetics topic, module 7. And today we are looking at dihybrid inheritance. So last lesson we looked at monohybrid inheritance, and today we're looking at what happens, what happens when more than one gene is inherited together. Okay, so to begin with, could you please pause me in a second, have a go at these questions. We're thinking about peas again because you know your man Mendel loved them. So how can we get so many different colours and textures of pea, such as those seen in that picture? If you're not too sure, have a guess, and we'll come back to it at the end of the lesson for you guys to check how much you understand. Okay, so pause now. And when you're ready, we'll play and we'll get started. Now, as I said, this is lesson 7.3, dihybrid inheritance. We're going to be looking at how two genes are inherited together and drawing those in a massive genetic diagram. Okay, so let's get in. Dihybrid inheritance occurs when genes are inherited together. Okay, so imagine I've got a chromosome like this, because we learnt about that last time, and I've got two gene loci, maybe down here and here. Okay, and maybe I'll say that the top one is for roundness and the bottom one is for colour. Now, these two genes are very close together, and this means that when things like crossing over happen in meiosis, those two genes aren't going to be split. This happens much more often than monohybrid inheritance. In fact, that monohybrid inheritance that we looked at last lesson is really rare. So it's very unlikely that the colour or the gene for colour is ever going to be just inherited by itself. Okay, in fact, what we've got is loads and loads, thousands and thousands of genes inherited together. Now, in terms of definitions, you need to know this one as well. The dihybrid inheritance is the inheritance of two genes, not two or more. We're only speaking about two genes, like a dimer for chemistry, things like that. Two genes at one time, but they're not necessarily on the same chromosome. So I gave you the example down here of them being on the same chromosome, but sometimes they just get inherited together randomly or for other spatial reasons. So imagine I've got these alleles for phenotype, okay? Big Y is for yellow seeds, little y is for green seeds, big R is for round seeds, and little r is for wrinkled seeds. Okay, notice that I haven't really followed my own instructions from last lesson, because I told you not to use letters that look very similar, uppercase and lowercase, but my writing is quite clear that one is bigger than the other. So could you please, using what you know, and even if you're having a guess, have a guess of what is the genotype for pea seeds which are homozygous yellow and homozygous round. And pause now. So hopefully you should have been able to recognise that for homozygous yellow, that first pea is going to have two big Ys. And the fact that it's homozygous yellow and homozygous round means that its genotype is going to have two alleles for the yellow, and we can say that it's homozygous dominant for colour, and two alleles for roundness. And notice how I've kept the Ys and the Rs together. That's really important. You don't want to be sitting there writing genotypes like this just yet with Ys and Rs combined. That's a bit of a mess. Okay, so hopefully now if you want to pause again and double check your answer for the second question, that would be great. Now for homozygous green seeds, the homozygous recessive ones, we're going to have two little Ys, and because they're wrinkled, they're going to have two little Rs. And again, I've made sure that I've got two alleles for colour and two alleles for the texture, the roundness or the wrinkledness of those peas. Okay, that's absolutely fantastic if you got those right. Well done. If you're not too sure, you can ask your teacher if it's me on Teams or whoever it is over your video call as well, and they should be able to help you in a little bit more detail. Okay, so now I've got two parents. I know their genotypes. I know that this doubly homozygous dominant P is yellow and round, and I know this doubly homozygous recessive P is green and wrinkled. Okay, now what I've got to do is I've now got to consider the gametes, because remember when we're doing a Punnett square, we start off with our parents. So I've got a yellow round P, and I've got a green wrinkled P, and I write out, highlighter, I write out what they look like underneath. So yellow, round, green, wrinkled. And we know that the genotype for that parent is Y, Y, R, R, big Y, big Y, big R, big R. And the green wrinkled one is little y, little y, little r, little r. Now, for the gametes, we have to make sure that a Y always goes with an R, because you're not going to inherit two alleles for the same gene in a sperm or an egg cell. Okay, now the easy way to do this with these homozygous genotypes is just to recognise that a big Y, let me do that in a different colour, make it easier to see, that a big Y is going to go with a big R for both of those. So gametes wise, we've got big Y, big R, big Y, big R, and likewise for those, the green and wrinkled gametes, you've got little y, little r, little y, little r again. Okay, so in terms of our gametes, where's my little hand? You're going to have from parent A, the yellow round one, two gametes that have got big Y, big R in them. So maybe they're the sperm. And in parent B, you've got two gametes that are little y, little r. Maybe I'm just going to draw those a horrible egg shape. Okay? Now we should be able to put that into a genetic cross like we have done previously. So if our parents are homozygous for both traits in their genotypes, like we've seen, we've got two big Ys and two big Rs, or two little Ys and two little Rs, then we just do a very simple Punnett square. So we saw from our previous slide that when we're looking at our gametes from these two homozygous parents, we've only got one type of gamete for each. So we know that for our yellow round parent that we've got the big Y, big R gametes, and for our green wrinkled P parent, we've got little y, little r's. And that's just going to go into a bog standard Punnett square. Okay, the same one that we did last time with the monohybrid inheritance. Okay, so could you have a go? And I'll give you a little hint that your Punnett square should start looking something like that. Okay, so pause now before you go on to the next slide and see if you can figure out the phenotypes and genotypes of the F1 generation when these two parents are crossed. Pause now. So now we're looking at the offspring of the cross that is up in the top left corner. Okay, we got two parents that are yellow and round and green and wrinkled, and we found out their gametes, and those are the ones that we drew in red, remember? Because we're following our rules. So we've got two sperms there, and then we've got two eggs. They look the same, but that's an egg there. Now what I've done is I've put the parental genotypes on the downwards, the side where the y-axis would be, and the maternal or female gametes at the top. It doesn't matter which side you do it on, really doesn't matter. But what we should have is a selection of offspring with the following gametes. Okay, remember when we write them, we keep the Ys and the Rs together. So this offspring is now big Y, big R, little r. And I just want to point out that you could have written it big R, little y, big Y, and you could have written it that way. That looks ugly. Okay, that looks really messy. So write the letters in the order that they come in. So the Y's come before the R's. And it's always better, just for in terms of like neatness sake and make it look like proper English, that the capital letters, if there are any, come first. Okay, so although this isn't technically wrong, writing little r, little y, big Y, it looks a bit messy, a little bit slapdash, like you're not too sure what's going on. And you will have noticed that all of those offspring have the same genotype. So they're all heterozygous for both traits. Hetero, so we've got a big letter and a little letter, a dominant and a recessive allele for each of those traits. Okay, when I say traits, I'm talking about characteristics, just in case you weren't sure. So when we're talking about the genotypes, 100% or 4 out of 4 offspring are big Y, big R, little r. And if we go back to our key down the bottom, maybe the information that we got from the stem of the question if we're in an exam situation, you will have been able to figure out that because there's a big Y and a little y, that means that the dominant trait is being seen. Okay, so we know that it's going to be yellow. And we've got a big R and a little r there. And again, that dominant trait is going to be seen. So all of our offspring are yellow and round. Okay, that's fantastic. So that is the basics of dihybrid inheritance. So this is what the Punnett square should look like, but they've made a little error when they've done the typing. And I want you to pause now and see if you can find where the error is in this Punnett square. Okay, I specifically said the Punnett square, take a look. And what you should have noticed is that there's probably been some kind of like autocorrect or something here, because these parent gametes from the green wrinkled seed shouldn't have the capital Y. They should actually be written lowercase y, lowercase r. Okay, so maybe when you're using your textbooks and stuff, double check, because this was just a screen grab taken from the textbook, double check that the Punnett square is actually correct. Okay, this was just a bit of a typo, but you can see in the offspring boxes, so the middle 4, that there needs to be a little y and a little r. Okay, which means that this parent over on the left hand side has had a little bit of an issue when they've been typing it out. But now we've got to think about what happens if we want to cross a parent that is big Y, big R, little r, with another one. So this time we're going to cross 2 of those F1 plants. Okay, and those were those big Y, big R, little r plants. And that gives us what we call the F2 generation. Right, so the second level of that family. Now, this is where it gets a little bit complicated, and I need you to bear with me because this is where it's going to start getting a little bit bigger. Imagine I've got big Y, big R, little r. Okay, the same genotype that we've gone through over the last couple of times. I know that I can't write 2 Y's together as a gamete or 2 R's together, because the Y's and the R's are inherited together but they're actually on separate chromosomes. Okay, so imagine I've got my chromosomes. I'm going to have one with a big Y and one with a little y. And at the same point, I might have one with a big R and one with a little r. Okay, it's 2 separate chromosomes. So it's actually not feasible biologically for the 2 Y's to get stuck together. Okay, so what I do instead is I've got to list the alleles using this very simple rule, 1, 3, 1, 4, 2, 3, 2, 4. And now I know your exams aren't going ahead, but hopefully you guys will be going to uni next year, and if you're doing anything biological, you might need to be able to do this if you're doing a genetics module. So the way that we do it is we label up our alleles 1, 2, 3 and 4 in the order that they're written. Now, as we said, we're going to label them 1, 2, 3 and 4, and all I have to do is stick the letters together in the order that that little rule goes in. So if I stick the letters 1 and 3 together, I'm going to get a gamete with big Y, big R. if I stick them 1 and 4 together, I've got a big Y and a little r. 2, 3, I've got little y and big R, and 2, 4, little y, little r. And you can see now that I have got 4 potential gametes from that single parent. And this is why the Punnett square gets a little bit bigger. When I say a little bit, I do mean quite a lot. Okay, now you can do the same with any other selection of gametes. So imagine I've got, think of a letter. So I've got F, or big F, little f for flowering, and we'll do the R's again. Big R, little r for round, whatever, it's just a silly example. 1, 3 is going to be FR, or big F, big R, and then 1, 4 is big F, little r. 2, 3 is little f, big R, and 2, 4 is little f, little r. Okay, follow the 1, 3, 1, 4, 2, 3, 2, 4 rule for parents that are heterozygous for at least one of those traits. Now, I would like you to think about this very carefully. I need you to pause. I want to cross 2 of those F1 plants, big Y, big R, little y, little r. And I would like you, please, to have a go at these questions. Okay? Big Y, big R, little y, little r, 2 plants. Can you please create a Punnett square, as we've done following all of the rules from last lesson? What are the phenotypic ratios? So I need X to Y to Z to A if there's 4 different phenotypes. How many different allele combinations are there? So you might need to have a look and count those. And then why is there a difference between your observed and expected numbers of each phenotype? Now this is going to be a little bit difficult. Question 4. Sorry, let me put the numbers in. 1, 2, 3, 4. Question 4 is going to be a little bit difficult if you didn't do the extra reading at the end of the monohybrid inheritance lesson when you looked at genetic, ratios and things like that. If you didn't, go back and do that before you carry on. So pause now, give yourself maybe 10 minutes to have a go at all of that, and then play when you're ready. Okay, so we're starting our Punnett square with our 2 parents that are yellow and round, big Y, big R, little r. And we know that each parent can give us the gametes of big Y, big R, big Y, little r, little y, big R, so little y, big R, and then little y, little r. And that occurs in both parents. So big Y, big R, big Y, little r, little y, big R, little y, little r. And then we're going to take those gametes, the sperms or the eggs, whichever one they are, and put them in a Punnett square. And this is where I said it gets a little bit more complicated, because each of those gametes needs to go on one side of a Punnett square. And now you've got a big 4x4 Punnett square. So Y, I'm going to put this first parent here and the other parent on the other side. So I'm going to put all of the gametes in that are possible, and I'm going to be really careful to make sure I'm writing them nice and visibly as capitals or lowercase, just so I don't get myself confused later on. And then I'm going to fill it out. Okay, so I can get rid of that column towards the end. Now remember, my Y's stay together and the R's stay together as well. And the easiest way I find to do it is to write them out, Y, or big Y, big R, little r, and using my key from the previous question, so I know that big Y is yellow, little y is green, big R is round, and little r is wrinkled. I find it easiest to write what they're going to look like in each box. So only very small. Make sure you can rub it out in an exam if you need to. But I know that big Y, big R is going to be yellow and round. Okay, and that helps me later on. Maybe I can colour code it. maybe I can do something else, whichever way you want to do it, but keep tabs on what each of them looks like. So this one is big Y, big Y, big R, little r, big Y, little y, big R, big R, big Y, little y, big R, little r. And I'm going to carry on doing that through the whole of my Punnett square. So I'm going to stop the video now and just finish filling it out. And when the recording plays again, it will magically have filled itself in with what all of the offspring look like. Okay, so now this Punnett square is done. It should have magically appeared for you. You can see that we've got every box filled in with the combination of Y's and R's. Notice that the Y's are stuck together and the R's are stuck together. Capital letters will always come before lowercase for each letter. So big Y always comes before a little y when you're writing it out. And notice that I've used a little key over on the right hand side to colour code my phenotypes, so I know what each box is going to look like if that offspring were to magically appear. Now I would say be careful writing anything else in your Punnett square box. So for example, where I've written yellow and round or yell/RO, it might be better for future to just draw a little dot and have a key. There's nothing stopping you taking coloured pens into an exam. I know you aren't doing them, but just for future reference, taking them into an exam is absolutely fine. Even if you're doing it online, so we might be giving you assessments soon, it's absolutely fine to use different colours as long as your answers are really clear and visible. Now, looking at this Punnett square, you should be able to pause and carry on answering those questions from the previous slide. So if you've forgotten what they are, pause the video, go back to the questions. I don't think I can flick between slides without causing major issues, and carry on. So this is what your offspring should have looked like. This is what the Punnett square would look like if it's nice and neat. And the phenotypic ratio is 9 to 3 to 3 to 1. 9 yellow round seeds, 3 yellow wrinkled seeds, 3 green round seed, and 1 green wrinkled seed. Now this 9 to 3 to 3 to 1 ratio is an example of the perfect Mendelian ratio. It's Mendelian inheritance. Okay? It's going to occur not very often in real life because of lots of various reasons, include things like crossing over and independent assortment, which we'll look at in a second. But you need to be able to recognise that term. The 9-3-3-1 ratio is perfect Mendelian inheritance. So there are 9 possible allele combinations in that Punnett square. If you're not too sure, go back and take a look at it. And now the reasons for differences between observed and expected. So let me just write up here. Differences between observed and expected phenotypic ratio are because of a few reasons that I've got listed here. So if you didn't get these or you weren't too sure over them, please make a note now, pause this video and make a note now of these possible reasons for the differences. So the fertilisation is random. It doesn't necessarily say that in every instance that big Y, big R is going to combine with the little y, little r. Okay, so an accurately calculated ratio might not actually occur as regularly if we've got a small sample size. Remember, if any of you do psychology or sociology or other human studies kind of subjects, a small sample size is generally a limiting factor for accuracy and reliability in every instance. Okay, so if you can't remember anything else, go for small sample size. However, if our genes are on the same chromosome, so like we've done in a few examples, we've got Y and then R just below it, if they're too close together, the genes are always going to be inherited. So when we do crossing over, like you should have learnt about in the meiosis lessons, if we've crossed these bits over between the dashed lines between two adjacent chromosomes, they're always going to be inherited together. And if you're not too sure about what I mean, ask your teacher or have a look online or go back to your Module 4 lessons from last year. Now, before we carry on and before you do some independent work, because, you know, you've got to do something at some point, have a go at figuring out or finding out what the law of independent assortment is. So pause, and when you're ready to carry on with the answer, play again. So when we're thinking about independent assortment, in terms of definition, it is where each member of a pair of alleles combines randomly with an allele from another pair. Okay, so in terms of independent assortment, we're talking about if I've got a cell with a couple of chromosomes undergoing meiosis. So imagine I've got this cell, it's a cell from a fly, and it's only got 2 chromosomes in it, and one arm of the chromosome is that way, and the other arm with a different set of, well, the same genes but different alleles is there, and then I've got another chromosome underneath it. We've got that one and that one. Okay, those cells, or those chromosomes, sorry, could line up like that. Alternatively, they could line up like this. And they could stay like that, or they could rotate themselves around either direction. Now, what that means in terms of divisions is that if I've got this cell on the left and it's going into metaphase 2, so maybe it's doing its second division, if the chromosomes are arranged as they are in this picture, then this gamete is going to have the combination of whatever genes are on the black arm and whatever genes are on the pink arm, and this cell will have whatever combination of alleles are on the blue arm and the green arm. Okay. Conversely, if we've got this cell, the second cell, the cell on the right hand side in metaphase 2, and they're dividing, then I've got different gametes this time. Okay, the left hand side of the cell is going to have the pink arm and the blue arm, and the right hand side of the cell is going to end up with the green arm and the black arm. Okay, and those chromosomes, or those alleles and the chromosomes in those cells, can now combine completely randomly with an egg that has got chromosomes in there that have lined up in a completely random way as well. And remember, this is going to be happening in every cell as it undergoes mitosis. Okay, so in your ovaries, in your testes, in the stamen and things like that for the plants, this is going to occur for every cell that is undergoing mitosis. So all of the gametes are completely random genetically. And that's really, really important to remember. That's a key source of variation from asexual reproduction or meiosis. Now we're going to have a go at some problem-solving questions. And we're going to do this first one together because it's a little bit complicated to use the words and recognise the information that you need to get from them. Okay, so a certain type of congenital deafness. Now congenital, if you don't know, means from birth. So this type of deafness that you get from birth in humans is caused by a rare autosomal dominant gene. Okay, so autosomal means that it's on a body chromosome or not sex-linked. Not sex-linked. And the fact that it's dominant means that we know automatically that deafness we can write with a capital D. Okay, and lowercase is going to be hearing. In a mating involving a deaf man and a deaf woman, could all of the children have normal hearing? And you need to explain why that's possible, or if it is possible at all. So normally in an exam, it would tell you whether the parents are homozygous or heterozygous, and in this case, they haven't actually said. So I'm going to assume that for an autosomal dominant condition, only one allele needs to be present for the phenotype to be present, and therefore I'm going to presume that the parents are both heterozygous. Okay, so I know that the mum is deaf. She's going to have the genotype big D, little d, and therefore the gametes are going to be as so. And likewise, the father, I don't know why I wrote father rather than dad, is deaf, big D, little d. and those are the gametes again. Now if I bung those gametes into a Punnett square, looks a little bit like that. And in terms of my offspring, I know that 25% are going to be big D, as you can see in the top left corner. 50% are big D, little d, over where I've circled them. And then finally we've got one little d. And we've got to use our key, our knowledge. We know that the deafness is dominant. We only need one allele, one big D allele for deafness to occur. So if I carry on thinking about those phenotypes now, both the big D and big D, little d offspring are going to be deaf, and the 25% little d are now hearing. And if we think back to the question, which was, is there any chance that all of the children could be hearing? There is no chance that all of the children could be hearing, as the deafness is a dominant condition. Now I would like you to use what we did in the dihybrid cross and have a go at this question. In Pokemon genetics, the genetic symbols are as follows. Big Y is a yellow body, little y is a white body, big T is a jagged tail, and little t is a straight tail. In the cross, big Y, big T, crossed with another big Y, big T, little t, what fraction, and notice I've been very specific with what format I want the answer in, of the F1 Pikachu will be, firstly, yellow, secondly, have a straight tail, either colour, be white with a straight tail, and yellow with a jagged tail. So pause now, have a go, start from the very beginning, do your Punnett square, work it all out, and come back when you are ready. So pause now, and then play when you are done. So this is going to be, again, a quick run through of how to do that Punnett square. If you're not sure, I'm going to start from the beginning. I know that I've got a parent that is yellow and has a jagged tail, and another parent that has a yellow colour and a jagged tail again. So phenotypically, they are yellow and jagged, yellow and jagged. And if you are confident in your Punnett square, just skip this part of the video. That's absolutely fine. I don't mind. Okay, just scroll on. This should take a few minutes, and on the next slide you should be able to see the answers. Now, following my 1-3-1-4-2-3-2-4 rule, we know that the gametes are going to be big Y, big T, big Y, little t, little y, big T, little y, little t for both parents. So remember, make sure that you're making your letters really obvious, really visible, and then we just bung those in the square. 1, 2, 3, 4. 1, 2, 3, 4. So big Y, big T, big Y, little t, little y, big T, little y, little t, and we go again down the side. Okay, now this time I know that I'm going to need a key because I'm interested in what they look like. So I'm going to make a little key for myself down in this left hand side. So the key, I'm going to have a little, I'm going to highlight the box in yellow if it's yellow, and I'm going to do a little jagged symbol if it's a jagged tail. So yellow, jagged. And then if it's white, I'm just going to leave the box blank, and I'm just going to do a straight line like that if it's got a straight tail. Okay, and now what I'm going to do is I'm going to go through each of my boxes, and I'm going to fill out the genotype and the phenotype using my symbols, okay, not using words in the boxes this time. Okay, remember we said it can start looking a little bit messy, so I'm going to pause my recording now. I'm going to fill out the table and play again when you are ready to see the answers. Okay, so you should be able to see now my completely filled in Punnett square, and I know that all of the ones that have got at least one big Y are going to be yellow. So that's yellow, 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 and the rest aren't. Okay, now thinking about my tails, I know that if I've got a big T, that it's jagged. So all of these top ones, jagged, 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 jagged. And all of the rest are straight-tailed. Okay, so these ones that have got the little lines are yellow straight-tailed. Now I've got to think about these white ones in the bottom corner. Okay, they've all got two little y's. They're homozygous recessive, so they're not going to have any colour. But some of them have got jagged tails. And one of them is white. This bottom right-hand one is both white and straight-tailed. Okay, so now I've got my key, I've got my colours, I know what's going on, hopefully, and this is where you should be before starting answering those questions. This slide, that we're all good. So for those of you that saw the previous section of video, you'll recognise this Punnett square. If not, then this is what your Punnett square should hopefully look like. Okay, notice that I've used the key down in the left-hand side of my own choosing. I've shaded the squares in yellow where the offspring would be yellow, and I've left them blank if they're white. And likewise, I've put a little jagged symbol if the tail is jagged, and I've done a little diagonal line if the tail is straight. Now, for the offspring to be yellow, they need to be either, they either need to be homozygous for the dominant Y or heterozygous. And you can see there in my Punnett square, they're the ones that I've shaded yellow. There are 12 of them. And this is why in certain questions it's actually easier to explain their phenotypes as you go along using colours and things like that. Because for this question, I know I can just count that there's 12 yellow boxes, 16 of them in total. So 12 out of 16 are yellow, which gives us around 3/4. Now for the straight tail, the T's need to be homozygous recessive. Okay, and for those, you can see, again, I've put my little symbols. I've got straight tails here, 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 and here. Okay, so 4 out of 16, or 1/4 of my offspring are straight-tailed. Now finally, penultimately, for the white and straight-tailed offspring, they need to be both homozygous recessive for the Y and homozygous recessive for the T. And that only occurs down in this bottom right-hand box. Okay, and the last is how many are homozygous, how many are yellow and a jagged tail. And now this is the bit where it comes in handy, having that key already done, the labelling of my phenotypes already done, because I can see that I've got 1, 2, 3, 4, 5, 6, 7, 8, 9 yellow jagged tail offspring. And I can't simplify that yellow jagged tail 9 out of 16 nice and easily. I could do it as a decimal, I suppose, but the question specifically asked for a fraction.